Dear students, I am Sanjeev Singh, GLA College Botany Department, and this is the third video on the topic DNA repair, which is in the syllabus of semester 3 Botany, NPU University. So now we have discussed base excision, nucleotide excision. Now we will be discussing mismatch repair. Now the first aspect in mismatch repair is whatever system is, whatever the cell is, it should know that the incorrect or the correct match. So how it is detected? So first I'll tell you the theory, then I'll show you the diagram and explain it in that. So in Escherichia coli, there is an enzyme known as DAM methylase. This enzyme is responsible for methylating, that means adding methyl group to all the adenines, that is the adenine base, which occurs within 5' guanine, adenosine, thymine, cytosine sequences, which begins from 5' So this particular enzyme methylates it, adds methyl group to the adenine. So how does it do that? So just immediately after DNA replication, the template strand means that strand which is to be copied into the new strand. This template strand is methylated and the new strand is not. So in this manner the template and the new strands can be distinguished, and this is how DAM methylase distinguishes it. So it is distinguished, then the thymine, the methyl groups are added to the newly synthesized the template strand. The template strands are methylated. Now to begin with, there are three most important proteins which are required, that is MUT-S protein, MUT-L protein and MUT-H proteins. They add in sequence. First MUT-S is initiated, then it activates MUT-L, then it activates MUT-H. Now MUT-S binds to mismatched base pair. This MUT-L binds to this complex, that is the MUT-S and the mismatched base complex, and activates MUT-H, which then binds to the GATC sequences. Now MUT-H cleaves unmethylated strand at the GATC site. Now the MUT-H starts cleaving the unmethylated strand because that is the new strand, and that has the mismatch is there and that needs to be corrected. Now the segment from cleavage site to mismatch is removed by exonuclease. Now that whole segment has to be removed because it is a mismatch. So this is removed by an enzyme called exonuclease. There are two types of enzymes, exonuclease and endonuclease. Exonuclease which cuts DNA from one end, and endonucleases are those which cuts DNA in between. Along with exonuclease, also required are helicase 2, and SSB proteins are also required. SSB protein, helicase, we know that it is the enzyme which uncoils the helix of the DNA, and SSB proteins are single strand binding proteins. These two are required along with exonuclease. Now when the cleavage has to begin from 3' site, then exonuclease 1 is involved, and if it begins from the 5' site, then exonuclease 7 is required. And then finally the gap is filled by DNA polymerase 3. In this case it is DNA polymerase 3 and not 1, and the DNA ligase. Now I'll try to explain this in the diagram. Now here you see 5' 3' end, this is the new strand. This is the template strand, 3' to 5'. So template strand are methylated. Fine. Now these three proteins begins their action. For MUT S will bind to this, then this MUT S will initiate MUT L, and this MUT L will then activate MUT H, and this will bind here and cause a cleavage here, the MUT H. So now with the help of exonuclease 7 single strand binding proteins, helicase 2, because the cleavage is happening here from the 5' end, so the exonuclease involved will be exonuclease 7 single strand binding protein and helicase 2. Now the gap is created over here because of that cleavage done by exonuclease 7, and then this gap is filled by DNA polymerase 3 and joined by DNA ligase, and that mismatch is corrected. Similarly, if it is happening from the 3' end, then you require exonuclease 1 single strand binding protein, helicase 2, and the gap is created here, just like the only difference is of the exonuclease 1, and then DNA polymerase 3 and DNA ligase joins it, and it is completed. So this completes the topic of DNA repair. I hope it must have been informative to you, and thank you.